Okay, so meiosis is going to be the sexual reproduction that occurs in cells. And this is going to obviously be in the sex cells then. In here is where we start to look at the heredity a little bit more, because that's where you're going to be actually passing on certain traits where you've got a possible combination of traits. With mitosis, what you get are little clones of the previous cell, so there's not really any variation. Here's where you can have variation. So heredity is just the transmission of traits from one generation to the next. And genetics is the study of heredity and the hereditary variation of things. So we know that offspring are going to acquire genes from their parents by inheriting the chromosomes. So the gene is going to be the actual unit of heredity, and it's going to carry the actual specific traits on there. It's going to be the instructions for those particular traits that a person will get. The gametes are going to be the reproductive cells that are in plants and animals. These are going to be the ones that actually transmit the genes from one generation to the next. It is your starter cell and the starter half of your genetics that you get from each parent in there. So gametes are those reproductive cells versus your somatic cells that are your non-sex cells, which are everything else in your body. And in humans, there's going to be 46 chromosomes in there. When we talk about the location of a specific gene, we call that the locus, and the plural term for that is loci in there. So when we compare sexual reproduction and asexual reproduction, the offspring is going to be an exact genetic copy of the parent, and you have a single individual as the sole parent. What you create are actually clones, genetically identical individuals. With sexual reproduction, it takes two parents in there to give rise to the offspring that will have their own unique combination of genes that they inherit from the two parents. So fertilization is going to be necessary for sexual life cycles in there. A life cycle is just the generation to generation sequence of stages that you'll go through in there. It's the reproductive history of the organism going from the time it's born to the time it makes its own offsprings. So when we look at the chromosomes, one of the ways to view them is a karyotype in there, which is an image of the chromosomes. And so you've got your chromosome here. This is a karyotype. There's actually websites out there that you can practice putting together karyotypes where they take pictures of the chromosomes, and this is what is done when they do a karyotype map on a person, is you'll get all of these individually like this and you match them up, but they're all random the way that you get the picture, and you match them up in there to see, is there an extra one or a duplication in there. And so the size of them is going to vary quite a bit in there, and they're arranged from the biggest ones down to the smallest ones. Right here is where you can see the X versus the Y chromosome, and the Y chromosome is significantly smaller in there. So they're arranged to be homologous chromosomes in there, where they're going to be paired with one that is the same length, the centromeres in the same region, and they have the same staining pattern because it's the same chromosome that is acquired from the other parent in there. The sex chromosome, which is your X and Y chromosomes, is going to determine the sex of the individual. So females are two X's, males are an X and a Y. And only a small part of it is homologous because of the significant size difference in there. All the other ones are going to be completely homologous. This one is not entirely homologous. So the non-sex chromosomes are referred to as your autosomes. So the cells are going to go through different stages in there depending on whether you're a plant or an animal, where you're going to be diploid or haploid. Diploid refers to having two chromosome sets in there, and that's how humans are going to spend most of their life, is diploid. Most animals are going to be diploid their entire life in there. Haploid cells are going to be the gametes that contain just a single set of chromosomes. In plants, it's a little bit different. You can actually have a multicellular gamete, and a lot of plants will go through what we call a gametophyte phase in there. But in the animal world, you're diploid all the way through. The only time you're a gamete is when you are a sperm or an egg. So fertilization is going to be the union of the gametes, and you're going to have the nuclei fuse. And once that happens, it becomes a zygote. The process of making the gametes is going to be meiosis. And in meiosis, you reduce the number of sets of chromosomes in there. So that's one of the big differences between meiosis and mitosis. In mitosis, you pass on and you have the same number of chromosomes. In meiosis, you're halving the number of chromosomes in there. So the sexual life cycle, there is some variation in there depending on what kind of organism you are. And the picture summarizes it real nicely down here. I know I'm going through this kind of fast, so if you guys want this fully typed in one, I'll put it up on Canvas. Yeah. Well, I like having the pictures in it better. It's a lot easier. You know, I would say that is a tool to use to study it, and I'll just give you this. Yeah. So when we look over here, you've got animals, and what you have are the gametes. This is your haploid stage, where you have one N is what they use to represent haploid in there. So you go through meiosis, you make these haploid cells, then it's fertilized. You have the zygote in there that's got a diploid stage all the way up through the multicellular adult organism that makes the gametes again. So the dominant phase for animals is going to be the diploid phase in there. In plants, they kind of do a 50-50 on things in here. So in plants, you've got this haploid gametophyte stage where they make the spores and your gametes, and then they go to fertilization, and they spend about half their life as the diploid in there. Some plants are going to be more dominant in one phase or another in there, but in plants you have multicellular in both phases. What is the difference in that? That's the difference that they look at, is where do you have your multicellular stage in there. And then with the fungi and the protists, they're at the other end of the spectrum. They will just have a zygote as your 2N stage. So the only time they are diploid is right after fertilization. Then they become haploid, and you have an either unicellular or multicellular organism that's going to spend most of its development and adult life as a haploid in there. So they are the other way of doing things in there. So that's the main thing you want to know, is which one. Humans, diploid most of the time. Plants, 50-50. Fungi and protists, haploid most of the time in there. Yep. Yeah, we're in there with the animals. Yeah, you'll get this exact thing that's up here. Yeah, and if you have a thumb drive with you, I can even put it on there before you go. So with meiosis, you're going to go through a phase of reducing the chromosomes from being diploid to haploid in there. So it goes through two sets of division. It's got the 4 phases in it that you have with mitosis. In there, you just repeat them twice. And in the first time, you'll copy the chromosomes. The second time, you don't. And that's why you end up with half as many. So you have your sister chromatids that are going to be held together with a cohesion in there. So you've got your 2 copies that are held together along their lengths, and so what it represents is one duplicated chromosome in there. One side is the same as the other side. They're just hooked together in the middle. So the homologous chromosomes are going to be that one that is the pair to it. The individual chromosomes that were inherited from the different parents will be your two homologous chromosomes. So you get chromosome 1 from mom, chromosome 1 from dad. And those two are homologous in there. So they're going to have different alleles for the gene. So you'll end up with a different possible trait that will be coded in the gene, but each of them is going to contain the same gene in the same spot. And the allele is just the version of the gene. So during interphase, before you do any division in there, you're going to start to pair the homologous chromosomes in the diploid parent cell. And then they're going to be duplicated in there. So this is where you're going to copy your genetic information before you get started. When it goes through meiosis 1, this is that first division that's almost exactly like mitosis in there, except you've got the genetic variation that can occur. You're going to have your homologous chromosomes separate in there, and you're going to have haploid cells with the duplicated chromosomes. In meiosis 2, you go through that division process again, and what you get is separation of the sister chromatids. So each of these only has one of the genes in there. So you start out with one cell and you end up with 4 instead of with 2 once you go through meiosis. So when you look at what goes on in meiosis versus mitosis, you're going to have prophase in both of them, and what's going to go on is the chromosomes are duplicated in there, in both of them, and you're going to have homologous chromosomes pair when you have meiosis in there. In metaphase, they both line up along the metaphase plate, and so it's metaphase 1 in meiosis. In anaphase and telophase, you separate to have your 2 daughter cells. So here the 2 daughter cells are each going to be 2N. In anaphase and telophase 1, you separate. You've got your 2 daughter cells here, but then they're going to go through meiosis 2, and that's how you end up with the 4 daughter cells, is because they divide one more time, and they just don't copy the chromosomes again. So things that are unique to meiosis, you have synapsis and crossing over. So the duplicate homologs pairs are going to form this synaptonemal complex and cross over. So they're going to bind with, they call the synaptonemal complex, where it binds with the homologous chromosome in there so that you can have switching. So when you inherit a gene or a chromosome from your parent, you're not necessarily going to inherit an exact copy of the chromosome they have. What's going to get passed on to you because of crossing over is they will make a new chromosome to pass on to you that is a combination from the 2 chromosomes they have. So you can have traits that they don't necessarily express get passed on to you because of that in there. The homologous pairs at the metaphase plate are going to line up as pairs of homologs rather than as individuals. And then you're going to have separation of homologs where the duplicate chromosomes of the homologous pairs separate. You don't have those pairs of homologs to separate in mitosis in there. So your sister chromatids are going to stay close together because the enzyme that is going to cleave the cohesin that holds them together is not going to be released until parts of anaphase 1 and 2 in there. So they're held together because they don't have the enzyme to break them apart until that point. And so a structure called the chiasmata is going to hold those homologs together And they're going to, as the spindle starts to form for the first mitotic division, and then when the cohesin is released, it allows the homologs to separate in there. So it's just waiting for that enzyme to come along to separate them. So we have reductional division versus equatorial division. With reductional division is what's going to occur in meiosis 1. It halves the number of chromosomes per set. The cell goes from being diploid to haploid as it splits the homologs, and you just have sister chromatids. The equatorial division in meiosis 2, the sister chromatids separate, producing haploid daughter cells in there. So where it gets more interesting with this is you can have a lot more genetic variation produced with sexual reproduction, which is why sexual reproduction, they say, will contribute quite a bit more to evolution in there, because it has more possible things that will be created out there to have better options be able to survive. So we have independent assortment of the chromosomes. So at metaphase 1, you've got your homolog pairs that are going to consist of one maternal and one paternal chromosome in there. And so there is a 50-50 chance that the particular daughter cell will get the maternal or the paternal chromosome. And it's completely independent for each chromosome in there, which one. So it isn't going to be, well, you know what, you got 12 of dads here, we need to give you 12 of moms here. You could end up with most of them coming from one parent, because there's a 50-50 chance for each individual chromosome out there. So occasionally you'll see where a child will just really strongly resemble one parent and looks nothing like the other side of the family. And that's how that can happen. They still have gotten some chromosomes from mom, but they inherited them much stronger on one side, from one side of the family, from one parent or one other. So the possible combinations for a human is going to be 2 times 2 to the 23rd in there. So each chromosome has 2 possible combinations in there, and then you've got 23 different chromosomes, so that's how you get 2 to the 23rd, which comes out to be 8.4 million possible combinations in there. So if you wanted to try for making 2 exact children in there and you didn't get twins, you better be prepared to try and make a whole lot of them. I mean, the idea of creating 8.4 million, it's like a suicide mission out there. Most people I know, they kind of max out at 15. So here it's showing you've got possibility 1, possibility that will occur, that you can have where you inherit this pattern, or you can get variation in there where you get some of the other ones in there. So within the individual chromosome itself, you can have crossing over. So this is where you're going to produce a recombinant chromosome in there, and it's going to carry genes derived from 2 different parents on there. So it's an average of 1 to 3 crossovers per chromosome pair. This will begin early in prophase 1 as the homologous chromosomes pair with the genes precisely lined up. So when they're lined up right next to each other, you get some of them that will trade places on there. So in the crossing over, the DNA of the sister chromatids is broken by specific proteins at precisely corresponding points to create a new combination. So where it's going to break in one is the exact point it will break in another, so that you don't try and fit a piece that doesn't fit in there. It makes 2 exact cuts on both sides so that you can swap the pieces on there. And so that's going to help create a lot more genetic variation. So you can see where you've got the non-sister chromatids that are held together during synapsis in here. the chiasmata, these additional places where it attaches. And in anaphase 1, you can see where the little piece of blue and red switched on here. That's the result of crossing over. So it'd be an average of 1 to 3 times that will happen on each particular chromosome. So on here, it's showing there being, it shows 2 on here, 2 on here. So when you go through anaphase 2, so you had crossing over occur for this one, but you didn't for this one. So when you go to make sperm or egg, it's possible that you could have one that has whole chromosome, one that has mostly your one type of chromosome, 2 bits of the other in there, or the other chromosome in there. So there's a lot more variation that occurs. And when you consider how many different sperm and egg you produce to have all of those different variations, it just makes a huge amount of variation in there. So then we have random fertilization in there. So in humans, each male and female gamete represents about 1 in 8.4 million possible combinations in there. And then your fusion of the zygote is going to be that 8.4 million times 8.4 million in there. So the 8.4 million is how many possible combinations you can make. This is how many possible combinations you can make with just one other person. You swap up the mate in there, and then you've got another huge variation of what you can create in there. So the odds of creating 2 exactly the same humans pretty much are so far against you that it's never going to happen. So if you decide you don't like husband number 1 and you want to try a kid with husband number 2, and you start with, you have your 8.4 million possible combinations and his 8.4. So now you've got your 8.4 and a different 8.4 million that you add in there that you can possibly create in there. So it just creates a huge amount of variation in there when you have different mating patterns in there. So where this is significant in evolution is natural selection is going to result in accumulation of the genetic variations favored by the natural environment. So one of the things that they think where we can look at this with humans is we have not always had easy access to food. So if you could store food efficiently and gain weight easily, that was a genetic advantage for a long time. We have not had that many generations of humans that can just drive up and get food, and you can probably eat plenty of food all day long and never get off your butt. So it's a whole different thing that would be a genetic advantage now, because what was a genetic advantage when food is rare is not a genetic advantage when food is abundant. And we're still hardwired to want to eat everything we find in there. So the genetic variation that was favored by the environment for so long was to gain weight. So if you gain weight easily, you were actually from a survivor back in the time of it being harder to get food. If you burned off that food really fast, you probably starved to death and never got to procreate out there. So that's how we can have these things get passed along. Mutation of the different alleles can occur along with the variation that you get with sexual reproduction, produces a huge genetic diversity out there. If you've got a more stable environment, asexual reproduction is more advantageous because it's going to perpetuate the successful combination of alleles. If the environment is less stable, you want to have sexual reproduction, not that you get to make any choice in this matter, but what would be more advantageous is to have more varieties in there so that you can potentially have something stronger adapt and survive in a population in there. With humans, we actually are really, really slow to adapt because we have so long between generations in there. So they figure the average generation time for a human, even with teen pregnancy, is 20 years in there. So your average of 20 before you start to produce offspring in there. If you're a cat and you're not fixed, it's less than a year before the first other kitty comes along that looks good and you start creating kittens in there. So with other animals, they will have more generations in there. So you can see the change in genetic traits much more quickly in species that reproduce more quickly in there. In the animal world, sexual reproduction is almost universal. The rotifers are one example that don't do sexual reproduction, but otherwise it is pretty much universal in there. We also haven't seen any that require 2 males to 1 female kind of ratio or anything like that. So the sexual reproduction is pretty universal in that it requires 1 male and 1 female in there. So horizontal gene transfer. When a rotifer dries up and its membrane cracks, it's going to allow DNA from other rotifers and other species to be able to come in through those cracks and be incorporated into the genome and increase its genetic variation. So they just pick up random bits of DNA from the environment. Bacteria can also do horizontal gene transfer. So one of the things with a lot of times people will go, well, I'm not worried. I haven't been on a lot of antibiotics in my life, so I'm not going to be resistant to anything. It's not you that is resistant. It's the bacteria in your body. So say you've never been on an antibiotic in your life. You were born sterile. All of those bacteria that you have in your body, which there's more of those cells than there are you cells, came from the external environment. They came from other places in there. So they could have all been exposed to somebody else who took a lot of antibiotics. If you eat meat, you most likely have eaten meat that has been exposed to a lot of antibiotics in there. So our livestock is given tremendous amount of antibiotics in this country. So to say that you haven't taken any antibiotics so you're not worried about acquiring an infection that way, you most likely have bacteria that are built from that pool that include those resistant things. Also, if you have one bacteria get in your body that has a gene for resistance in there, bacteria can actually duplicate that gene, and through horizontal gene transfer, they will make what they call a sex pilus that pokes the neighbor. Doesn't matter if it's the same species or not. And through that little pokey pilus that they make, it slides that gene into their body too. So they don't seem to mind. So the idea that, you look at it and go, well, the antibiotic resistance is somebody else's problem. No, it's all of our problem. And a lot of times in healthcare, people for years it's been an easy way to shut a patient up. One of them that comes in, and we will all get to deal with them out there, people that come in that are just needy, that they really just want attention more than something else being wrong with them. And they're very persistent. So they want to feel like something is being done for them. It doesn't really matter what it is, as long as it's something being done for them. So what had happened for a long time is, rather than arguing with them, you said, oh yes, this is the worst infection I've ever seen in my life, to indulge their ego in that. You wrote them a prescription for antibiotics and you sent them on their way. They didn't need it, but they figured there was no harm done. Now we know better. There was a lot of harm done in there. And so it's the horizontal gene transfer is a big contributor to a lot of the problems with resistance like that we have. So let's go ahead and take a break on this for a minute. The gene idea. So we've looked at how we create the gametes and the chromosomes to pass things on. But now once we have the genes in there, there was some different ideas as to how we came up with things. The first idea was this blending hypothesis. Back in the 1800s, They thought that the genetic material was mixed from the two parents, similar to the way the paint colors do. So thinking if you maybe had one with blue eyes and one with brown eyes, you were going to get something in the middle. And then some people looked at it a little bit more and was like, well, this doesn't completely explain it here. That doesn't necessarily work that way. So then there was the particulate hypothesis, which is more similar to the gene idea. The parents just pass discrete inheritable units that retain their separate identities into their offspring. There were quite a few people who worked on this, but Mendel is the best known in there. And I didn't do a lot with putting in people in history into this for you because there's enough other stuff to memorize without making it a history thing. If you're interested in the history of it, you go read it in a different context. So Mendel's experiment, he looked at pea plants for his, and that determined that a character was a heritable feature that varies among individuals, such as flower color. So the different colors would be the different characters. And the trait is each variant for the character in there. So you could have a pink trait versus a purple trait versus a white trait versus a red trait in these things. And he chose, when he studied this, to track only characters that occurred in two distinct alternatives. So what he used were pea plants that produce flowers. And he chose some that had purple and white. There was no other variants that came in there. There was no pink or any other color combinations. And that was fortunate that he studied it that way because we may have had a whole different hypothesis and not have near the understanding of genetics we do now if it wouldn't have been choosing something that had just two distinct traits. So when we talk about true breeding, that's going to be using varieties that only produce the same variety as the parent plant after self-pollination in successive generations. So you would have plants that would only produce purple when they were bred with other plants that produced purple in there, and then you would have some that would only produce white variants in there. The hybrids are going to be the mating or crossing of two contrasting true breeding plants. With the true breed, you knew what their genetic trait was for there, because there was only one possible outcome. When you create a hybrid, then you don't necessarily know what its genetic trait is. So the P generation, or the parental generation, is the true breeding parents. The F1 generation, which the F comes from filial, which means sons in there. So the first son generation in here is the hybrid offspring from crossing the two true breed parents in here. And then the F2 allows the hybrids to self-pollinate to create an F2. So your sons that you created from F1 would breed with each other to make the F2 generation in there. So with your true breed parents, you had the purple and the white. The purple was the dominant trait, and so when you crossed a purple with a white, you got a purple. So your next generation, you had all purples in there. When you crossed those purples together, you got a combination of some that were purple and some that were white in there. No, plants, animals, bacteria, everything has chromosomes.
Okay, so these are kind of like from the, you said the two first sons would breed. Would they end up having, how come they don't have mutations like if either of you were extinction free? You know what I mean?
Well, they were pea plants. And they only looked at this trait in the pea plant. So that's... It usually takes multiple generations of inbreeding before the real problems start to happen. So if you look at a lot of like the British royalty, they were pretty bad for it in there. You saw that. But with the plants, with these, they didn't look at any other traits at the same time either. So you had these purple flowers here. Although they looked purple, the reason you have these white ones here that occurred is because some of them carried that gene trait for the white flowers, even though it wasn't expressed. And that's how you ended up with this mix of purple and white. But when you do the Punnett squares in there and you go through and you calculate some of those out in 15, you'll start to see how the odds create numbers that look like this in there. So with the law of segregation, if the blending model was correct, when you cross those purple and white flowers, you would have had a pale purple instead, but that's not what we got. What actually happened is you got the purple flowers, and then when you had the F1 generation self-pollinate, the white trait appeared in there. So you can have recessive traits that you would inherit from one of your parents that you don't express that trait, but you can pass it on to your child. And that's what the white is one of those recessive traits that was passed on. The dominant trait, the dominant trait is going to be the one that if you've got one of each is going to show up. The ratio of dominant traits to recessive traits occurs in about a 3-to-1 ratio in the F2 generation in there. And this was what Mendel used as his basis for deducing the law of segregation in there. So with the allele, it's the alternate versions of the gene, and what the law of segregation is going to state is that alternate versions of the genes account for the variations in the inherited characteristics. You get the white version or the purple version of the gene. For each character, an organism inherits 2 copies of the gene, one from each parent in there. If the 2 alleles at the locus differ, the dominant allele is going to determine the appearance, and the recessive trait has no appearance on the organism. So if you inherited a white and a purple, you're going to show the purple. You just carry a gene for the white, but you don't express it in there. The 2 alleles for a heritable character segregate during gamete formation and end up in separate gametes in there. So when you look at your P generation in there, the ones that had purple flowers, we represent them with capital letters for the dominant trait, small letters for the recessive trait in there. These had 2 big P traits in there for purple. These had 2 small p traits that allowed them to be white. So in the F1 generation, what you ended up with is they got 1 trait from mom, 1 trait from dad. So big P, small p, but they're going to appear purple in there. In the second generation in there, when you create this Punnett square that allows you to predict this, you put the 2 possible traits across the top from one parent, the 2 possible traits from the other parent down the side, and what you get, you take the top one here, the side one here, and you put both of those together. You get 2 big P's. You're going to have a purple flower. Over here, you get 1 purple and 1 white. So 1 big P, 1 small p, you get a purple flower. Same thing here. 1 big P, 1 small p, you get a purple flower. In this 4th box, you have 2 small p's. So does everybody understand how you get the things in the box? Okay. So you have 3, when you look at this, this is what your odds are, 3-to-1 of them being purple in there. You only get 1 mate at a time, Steve. Yeah, so what it's showing is if you have that recessive trait in there, that the recessive trait only shows up in the one that inherits the recessive gene for both parents. 2 of them are carriers of the recessive trait, and 1 of them doesn't have it at all in there. So it creates 1 other true breed. The thing is when you look at these 3 plants on the outside, unless you can identify which genes they have, you can't tell which one is which. So the Punnett square is the diagrammatic square for predicting the allele composition from the offspring in there with individuals with known genetic makeup. If you don't know their genetic makeup, you have to do a Punnett square for each possible genetic makeup. And once those patterns have been produced in there, you know what the numbers are, the odds in there, so then you don't make the square anymore. But the square was how the numbers were originally determined in there. So with this one, it's saying, okay, we have a purple flower here. We don't know what its genetic makeup is. We know it can be the 2 big P's or 1 big P, 1 small p. When we made it with the white flower, we know the white flower has to have 2 small p's. So we have to do 2 Punnett squares to figure out the possibilities in there. So if it's mated with the white flower, and this is your normal F1 that you would get from the true breed in there. And with the other one, now you get the 50-50 ratio in there. So you can see how those are put together. So any questions on how those boxes? You will do some Punnett square boxes. That's what you're doing in Lab 15, is filling in Punnett squares on there. What? What about it? Yes. Yeah. Yeah, once you make a few of them, as long as you'll look at an example and you get it, once you do a few, you're like, okay, got it, easy on there. So some other genetic vocabulary in here. If it's homozygous, it means that the organism has 2 identical pairs of alleles for the character. So they're going to have the same type of gene in there. Heterozygous means they get 2 different alleles for the character. The phenotype is what is actually expressed or observed in the organism, so what you actually see for the color of the flowers. The genotype is the genetic makeup. So when you have an unknown genotype in there, you can do a test cross with them, breed the organism with a recessive homozygote in there, and based on do you get this pattern or this pattern in here, then you can determine what the genetic pattern was of the parent. So that's great in pea plants and everything, but not really very practical if you look at a human and you want to go, do I really have both traits for brown eyes or do I have a trait for blue eyes? You don't go just making a baby just to see what comes out on that. So pea plants, no big deal. Animals, yeah, I think it's a little more complicated in life. So with the law of independent assortment in there, you're going to have your monohybrids where the parents are heterozygous for one particular character being followed in the cross. And the monohybrid cross is that cross between the heterozygous for a single character. So the parents themselves are called the monohybrids, and when you breed them, we call it a monohybrid cross. So the cross part is the breeding part in there. So the second law of inheritance is identified by following 2 characters at the same time. So you can do these Punnett squares where you have 2 hybrids in there. So a dihybrid, the F1 plants are heterozygous for the 2 following characters on the cross. So for these, it's using yellow versus green and wrinkly versus smooth in there, and they're using Y and R as the letters. Again, Y is the capital letters are the dominant, the small letters are the recessive in there. So when you do a dihybrid cross, you're crossing between 2 F1 dihybrids, and the F2 generation is going to have a ratio of 9 to 3 to 3 to 1. And so this law is only going to apply to genes located on different chromosomes that are not homologous. When you're looking at things that are right next to each other on the chromosome, it can be thrown off and a little bit different from there. So when you do the dihybrid cross here, you've got your traits here. YR, big Y, big R, small y, small r. So when you cross those, the F1 generation is going to be yellow and smooth in there, but it's going to be heterozygous. It's going to carry one of each gene. So the dominant one shows up. So when you do the prediction for the first one in there, it's one half of these, one half of these. You get this 3-to-1. So this would be if you had dependent assortment, which is not what we have. We have independent assortment. So you won't make this kind of box. Instead, you would make this one. So what you would put is the sperm from each of these, the possible combinations you could have of the sperm. You could have a big Y, a big R. You could have a big Y, a small r. You could have a small y, a big R, and a big Y, or a small y and a small r. The same possible combinations for the eggs. So what you put in the box is all 4 letters from each of these. So here you have the 2 big Y's, 1 big R, 1 small r. So you end up with all 4 letters in the box in there, and you have 16. You always write the capital letter before the, well, you mean the order that you put these in?
Yeah.
It doesn't matter what order, because you're looking at all the possible combinations. And then what we do is we look at the ratio of how many of them got.
And so then the big Y and the little r, we're putting the capital first, and then the lowercase, and then moving on to the other r.
Yeah, so it's easiest if you do, like if you decide you're going to put Y first, you put Y first in all of it.
Okay.
If you decide you're going to put R first, you put R first in all of it.
Before.
Well, capitals, down here you usually write capital first in this box, but it doesn't really matter, because when you look at it, if you see a small letter and a big letter, you know that the big letter is going to be the observed trait because it's the dominant one. But the whole reason you create one of these boxes is so you look and you go, okay, how many are yellow and smooth? How many are green and smooth? How many are yellow and wrinkly? How many are green and wrinkly? Yeah, so then you come up with a ratio in there. And that's the whole purpose of doing the box. So if you wanted to say, well, okay, I've got these 2 plants and I want to breed this, how many am I going to get that are like this? You can predict with the ratio in there. So when we use probability, it ranges from 0 to 1 in there. So when we talk about events that are certain not to happen, they're assigned a probability of 0. Ones that are certain to happen are assigned a probability of 1. So when you add up all of the probabilities of something happening, that total needs to come out to equal 1. So your probability of getting the green or the yellow and smooth is 9 and 16. Green and smooth is 3 and 16. Yellow wrinkly is 3 and 16. And 1 and 16 for the green wrinkly. So if we add all of those up, it comes out to be 16 out of 16, which is 1. So the total of all of the probabilities. So your probability for this one is, this green smooth one is 3 in 16. It's between 0 and 1 in there. So we have multiplication and addition rules that apply to the monohybrid crosses in there. So the multiplication rule, to determine the probability of 2 independent events occurring together, you just multiply the probabilities together. So if you've got a probability of 1/2 of getting this and 1/2 of getting this, it comes out to be 1/4 in there. The addition rule, the probability that any one of 2 or more mutually exclusive events will occur is calculated by adding their individual probabilities. So if, for example, in there we have the probability for one possible way of attaining an F2 heterozygote, the dominant allele from the egg and recessive allele from the sperm is 1 in 4. The probability for it being the opposite combination is also 1 in 4. So with the addition rule, you add 1/4 plus 1/4 is 1/2 in there. So for solving more complex genetic problems in there, you can use the rule of probabilities to predict the outcomes. So if you were to create one of those 16 square crosses in there, you could predict the probability of getting any of the combinations in there. So then if you wanted to look at it and say, well, I want to have a chance of getting, this one looks at it doing 3 in there, because you can make these Punnett squares with 3, 4, 5, 6 traits in there, but then you get a really big square and it gets really tedious to fill all of that in and count that out. So you can use these rules of probability to predict things in there. So your probability of getting YYRR, 1/4 to get the double capital Y, 1/4 to get the double capital R. So when you do the rule of multiplication, it's a 1/16 chance of getting both of those together. Probability of getting the 1 big Y, 1 small y, and 1 big R and 1 capital R, for the Y component is 1 in 2. For the R component is 1 in 4. Now you have a 1/8 probability. So you can go through and look at it for getting, you can just continue that on with the addition or multiplication rules for doing 3 in there. So you use the multiplication when they're independent events in there and you're looking at saying, okay, I want to get my odds of getting, like here where it's saying you're looking at the probability of getting both dominant traits in there. So with that one, you're doing multiplication in there. Yeah, the addition rule is not used as often as the multiplication rule. So you could actually extrapolate this out for multiple traits if you wanted to. However, with what we know with genetics, this is used more as a learning tool for simple Mendelian genetics. But the big picture of genetics is actually a lot more complicated than, we're a lot more complicated than peas. So most of our traits, you don't have just 2 options in there. So this is looking at examples of really simple 2-option traits in there. So inheritance patterns, this thing, are often more complex than predicted by simple Mendelian genetics. So if you extend it on here, it's going to deviate from these simple patterns when the alleles are not completely dominant or not completely recessive. So these, if this was what they looked at first, they would have thought that there really was a blending hypothesis. And this is probably the reason that the blending hypothesis was even created in the beginning. So you can have incomplete dominance in there. So with complete dominance, the F1 offspring is always going to look like one of the 2 parents. The trait is either dominant or recessive. You get it or you don't in there. With incomplete dominance, you're going to have a phenotype that's somewhere between the 2 parental varieties. So these were done using red and white flowers in here. So your red ones, we're going to have the red trait, and the white ones, the W trait in there. So when you're using the dominant and recessive, you use the small letters and the capital letters to indicate that. When you're looking at blending, that is not necessarily done in there. It makes it too confusing. So usually with the blending, they'll do kind of this superscript form in there, or just use these letters up here for the blending of the traits. So you don't get confused when you look at it and go, okay, these are blended traits, these are dominant recessive traits in there. So your F1 generation, you got the pink flowers. However, when you crossed a pink flower with a pink flower, you still had these separate genes that are in there. What you get are one that's going to be a red flower, two that are going to be pink flowers, one that will be a white flower in there. So that's what your ratio ends up becoming in there. So codominance is another variation. We have this with our blood cells in there, with the human blood types. And so with this one, you're going to, there is the four possible blood types: type A, type B, type AB, and O. Type A is going to have the genotype marker for A. It could also have the O marker, which doesn't have anything. O actually means not a marker in there. So if you're type A, you got an A marker from both parents, or an A from one and O from the other. So all that is expressed is the A, because the O didn't have a marker if you got that type. If you got B, you got B marker from both parents, or B from one, O from the other. If you're AB, you got A from one parent, B from the other. And if you're O, you got O from both parents in there. So when we look at these between dominant and recessive, the allele that is dominant is going to be seen in the phenotype in there. So it doesn't interact with or suppress the recessive gene. That recessive gene is still there, it's just not being expressed in there. So some examples of diseases where this can occur: one is Tay-Sachs disease. Here the brain cells cannot metabolize a certain type of lipid because the enzyme doesn't work properly. So the lipids build up in the brain, leads to seizures, blindness, motor and mental difficulties in there. So at the organism level in here, the children with two copies of the allele have the disease. So you have to inherit two recessive traits in there, one from each parent in there. They'll have the traits. If you are a heterozygous individual, what's going to happen with there is it's an intermediate level, because this is somewhat codominant in there. So this person did not inherit, the person with the two recessive traits didn't inherit a gene that can make that at all. The heterozygous one inherited one gene that works and one that doesn't. So what they actually end up with is incomplete dominance at the biochemical level in there. They produce some, but not as much as another individual. So at the molecular level, the heterozygous individuals produce an equal number of normal and dysfunctional enzyme molecules, but it's enough to keep them functioning. They don't make as much as a normal person, but they don't manifest this disease because they make enough. So most genes are going to actually exist in more than two allelic forms. So your blood type is an example. There is the ABO possibilities in there. Pleiotropy is going to be where most genes have multiple phenotypic effects. So you've got pleiotropic alleles responsible for multiple symptoms that are going to go along with diseases like cystic fibrosis and sickle cell disease in humans in there. So you don't just have two options of how that gene is going to be expressed. So when you have Mendelian genetics that are going to apply to two or more genes, we can have epistasis in there, where the phenotypic expression of a gene at one locus is going to alter the gene at another locus in there. So the effect of one gene is going to control another gene in there. So one place where we see this is with lab dogs. So they come in black, brown, and yellow labs in there. And when you breed labs, you can breed 2 black labs, and it doesn't mean you're going to get a black lab. The litter comes out with a variety of puppies in there. And this will explain why in there. So the black coat is dominant to the brown coat. So big B is the black coat, small b is the brown coat. So in order to be a chocolate lab, you have to get 2 B's in there. There's a second gene involved that's going to determine whether or not to deposit the pigment, regardless of what color. So if you're a yellow lab, you don't deposit pigment. You can still carry genes for brown coats and black coats, you just don't deposit the pigment in there. If you're a black or a brown lab, you have the gene that says deposit your pigment in there. So the color is actually determined by these 2 genes in there. So if the lab is BB and the second locus is EE, it's going to be a yellow coat regardless of what color gene it gets for the pigment, because it just doesn't deposit it in there. So when you look at the numbers on this, you get 9 black labs, 3 brown, 4 yellow in there is the way that the Punnett square works out for that. So is that making some sense, the idea that one gene can control or influence another one? Yeah, it's just what here, it will, the B is going to determine, does it carry genes for a brown or black coat? The E is going to determine, is it going to deposit the pigment in its coat or not? So if it does deposit pigment, then it's going to be whichever is dominant and whichever is recessive pattern in there. If it doesn't deposit the pigment, it doesn't matter what the genes are, it's not going to deposit the pigment, and it's going to look yellow. Capital B for black, small b for brown. Yeah. So polygenic inheritance is the additive effects of 2 or more genes on a single phenotypic character, which is the opposite of pleiotropy in there, or the converse of pleiotropy. So this is looking at it with light skin and dark skin in here with sperm in there. So for example, we have some human traits like skin color and height that are going to be impossible to actually be able to do normal Punnett squares on, like the examples we've had so far, because they're going to occur in gradations in the population. And so with this one, with the colors, you can see in there, based on the combination, that you have totally dark to totally light, and you have all possible traits in between. So you can see this in humans a lot. When you have a parent that has really dark skin and a parent that has really light skin, you're not necessarily going to get a baby that's somewhere in the middle. You could have the baby come out really light or really dark or any variation in between. You don't know what you're going to get because they're all possibilities. You're not going to produce that kind of a square. The idea that you can have the gradations is, yeah. So nature versus nurture. Can the environment impact what you're going to see for the phenotype? Yes, it can. So This is another exception to the simple Mendelian genetics with the simple Punnett squares. The norm of reaction is the genotype is generally not associated with a strictly defined phenotype, but rather a range of phenotypic possibilities due to environmental influence. So those are the normal possibilities that can occur based on what the environment is. With what was that? Yeah, what are the normal possible range of things that you could find, provided you don't somehow do some strange thing to manipulate the situation. So the multifactorial characters, these are going to have many factors, both genetical and environmental, that are going to influence what you get. And so when you integrate a Mendelian view of heredity and variation in there, you've got genotype and phenotype that are going to refer to an organism in its entirety, not just specific alleles. So when we look at traits that a human gets, there is a lot more to it than just looking at the straight Mendelian genetics in there. The phenotype, that what you see on the outside, represents what that person's genes are and their unique environmental history. So this is why you can have identical twins and have one of those twins have a genetic disease and not the other one. There can be things in their environment that will trigger and control those genes. One twin may be exposed to that and the other one not in there. So with humans, the genetic span is too long for genetic research in there, so it takes about 20 years. So to look and see, well, okay, I want to know what kind of trait I have. Say you're 6 years old and you decide you want to know what kind of trait you have. You got to wait a while to do a test cross on there as a human. And test crossing humans to see just what you produce isn't the way we do things in there. That is not how we breed. So instead what we'll do is make pedigrees where you'll look at a collection of information about the family history for a trait and look back at history and see what the possibilities are that you have, rather than breeding to see what comes out in there. And so when you create one of these, which you'll be doing one of these in your lab book as well, males are squares, females are rounds. If you are affected by a trait in there, you color it in. When there is a mating, you draw the line between them. And this is where when they make comments like your genetic tree crosses or your family tree crosses, you know, you shouldn't have parents making offspring and then to put like a mating between those two. That's wrong. That's your inbreeding in there. So when you make one of these for yourself, unless you know that that's occurred in your family, if you're seeing lines like that occur between brother and sister, you've gone wrong somewhere. So then the offspring are attached by these lines. So the mating is the line in between. Offspring have the line up the top and come down to them. So one of the traits it looks at is a widow's peak, which is the pointy hairline here in the front, in there, and looking to see who in your family has a widow's peak going back. Another one you can look at is the attached earlobe. Does it come down and it's attached all the way here, or do you have earlobes that flap back and forth in there? Okay, so we're almost done with this one. So recessively inherited disorders. The heterozygous individuals for these, so the people who are carrying the gene for these, are usually normal in phenotype because they have one normal allele in there that will make enough of the specific protein they need that the disorder doesn't show up in them. It will only show up in the homozygous individuals with the recessive allele. A carrier is a person who is heterozygous. They carry that gene, they're just not expressing that gene, or the disorder is not manifested as their phenotype in there. Generally, you're not going to see genetic disorders evenly distributed among all groups of people, And a lot of that is due to different genetic histories in times when people were more genetically isolated. So you will see them becoming more spread out over time, likely, but there are certain disorders that you saw being spread in certain populations because you didn't see people going from South America to Eastern Europe to live. It was just not the kind of thing where you had a lot of cross-mating in there. It's long distance to travel. People don't generally move around that far away to different countries to breed like that. Contiguous matings, these are inbreeding, and so it's indicated in a pedigree by a double line. So the probability of passing on recessive traits increases greatly. And you see this a lot with domesticated animals. So a lot of times dogs and cats, particularly common breeds, say like a golden retriever, people have bred brothers and sisters and cousins together like that because with a dog, they don't really care. You can have the Chihuahua is more than happy to give it a shot with the Great Dane in there. They just don't care. When the hormone's there, they're ready to go. So the animals just don't know any better in doing that. But if you were to do that in humans, you're going to have much greater probability of diseases being passed on. So cystic fibrosis, this is one of the most common lethal genetic diseases in the U.S. It's in 1 in 25,000 people of European descent. It's more rare in other groups. About 4% of people who are of European descent are carriers for it. So the normal protein in here is, the normal allele is going to code for a protein that transports the chloride ion between certain cells. So this chloride ion in there is going to be what's involved with the thickness of making mucus and secretions in there. It's going to affect how thick they are in there. So people who have this trait, that channel is defective in there, and they end up with abnormally thick mucus that's more sticky, and it will build up in places like the lungs, the pancreas, the gastrointestinal tract, and other organs in there, which results in poor absorption of nutrients, chronic bronchitis, recurrent infections. It used to be if you had cystic fibrosis, you would not make it to adulthood in there. But now a lot of them will make it into their 20s and 30s in there. They may have to have lung transplants or other organ transplants along the way, but their lifespan is quite a bit longer. So I know one person who has it who is almost 40. He just got a lung transplant about a year ago and is actually doing pretty well at this point. So with sickle cell disease, this is the most common inherited disorder in people of African descent. And so it's about 1 in 400 African Americans. And you substitute a single amino acid in hemoglobin. So a homozygous individual is going to have all of their red blood cells affected. And when they're under low oxygen content, the red blood cells will start to aggregate into a sickle shape and clump. It clogs up the small vessels in there, and so they're going to have symptoms that are the result of not being able to transport the blood well in those areas, including weakness, pain, damage, and you can end up with paralysis in there if you're not able to get blood to those nerves. A heterozygous carrier, these will be people that they say have sickle cell trait. They're usually healthy, but they may manifest the symptoms if they're in a prolonged stress situation or with prolonged reduced oxygen in there. So they have some of their cells that will be disordered in there. So about 1 in 10 African Americans actually have the sickle cell trait. Your heterozygous individuals, one of the reasons why this is so abundant in there is they have a reduced frequency and intensity of malaria. So it was actually an advantage to be sickle cell trait in sub-Saharan Africa where malaria is abundant, and people that were completely susceptible to malaria had a much higher death rate of it. So it was a trait that actually got passed on and became more and more common because that allowed them to survive better in there. So dominantly inherited disorders, there aren't a whole lot of these. Achondroplasia, dwarfism, is in 1 in 25,000 people. So people who are heterozygous will have the dwarf phenotype in there. The dominant allele for a lethal disease is much less common than recessive diseases, because if it's a lethal disease, it's usually going to kill the person before they have a chance to reproduce. Huntington's disease, this is an example. It's a late onset lethal disease in there that's dominant in there, but a lot of times the person has already reproduced by the time they're aware that they have this in there. So it's a degenerative disease of the nervous system, and the person has no phenotypic effects until they're 35 to 45 years old. And by that point, you've usually already had your children and passed it on. So this affects 1 in 10,000 in the US. They can do genetic testing for this now if it runs in your family to check for it. However, there are some people out there that they may know they're a carrier of this disease, and they'll reproduce anyway and pass it on. So multifactorial disorders in there. You know what? Well, let's go ahead and stop there today rather than starting a new idea, since we only have 5 minutes.